In this video, we will discuss about the mitochondrial OXPHOS. It gets activated when there is a mutation in this SETD2 proteins in the pancreatic cancer cells. So, in order to in order to come to a conclusion that how they find out the relationship between mitochondrial OXPHOS and SETD2 mutations, they check the already available data and look for this SETD2 mutated cells and what are the metabolic changes that occurs in those kind of patients. And what before going into that analysis, they first check this whether this SETD2 mutations has any role with this TP53 or not. And what they have find out is that this SETD2 mutations occurs in the patients regardless whether they have any TP53 mutations or not. So in order to come to the conclusions, they compare the data for this 178 patients, and what they find out is that around 24 cases has both this TP53 wild type and SETD2 wild type, whereas 90 cases has TP53 mutations and SETD2 wild type, and 3 patients has TP53 wild type and SETD2 mutants. So it is not necessary that SETD2 mutations will also cause TP53 mutations. And another thing that they identified or did the analysis on these patients is that what are the pathways that are enriched in this SETD2 mutated conditions. And what they have find out is that in the case where SETD2 is mutated, then they are able to see oxidative phosphorylations are upregulated, and other pathways also upregulated, but the highest upregulations they are seen in the oxidative phosphorylations. Whereas in the case where SETD2 wild type is there, they do not see a significant increase in the oxidative phosphorylations. And same things they studied, they did it on this QCMG PAD patients from 96 patients, and they find the similar results. That is, when the SETD2 mutations occur, they see the enrichment of these oxidative phosphorylations, whereas not for the case where SETD2 wild type is there and TP53 mutations are there. And after finding out that oxidative phosphorylations has a relationship between SETD2 mutations, they did one more analysis of the patients that has SETD2 low and SETD2 high, and in this case also they find the similar results, that the oxidative phosphorylations are high or enriched for the SETD2 low conditions, both for this TGCA PAD and QCMG PAD. It can be seen here. So after confirming that the oxidative phosphorylations has inverse relationship with the SETD2 wild type or SETD2 mutations, that is, when the SETD2 is mutated, then there is upregulation in the oxidative phosphorylations. And in order to further analyze the other metabolic process that get affected, they did the unbiased single cell RNA-seq data on the mouse, which is treated with this this pancreatic cancer. And what they have find out is that first, using in the UMAP data, they are able to see 6 clusters that can be seen in this panel D. 1 2 3 4 5 6 1 2 3 4 5 6. These 6 clusters has been identified, and those cluster names are shown in the here. And after this cluster has been identified, they subcluster this acinar and ductal cells, that is, this acinar and ductal cells into 5 different subsets that can be seen here. And among these 5 subsets, more than 50% of the populations are occupied by acinar cell and ADM. ADM is the acinar ductal metaplasia. It is the combination of both the acinar cell and the ductal cell. And since we see that around 50% of the populations are occupied by the acinar and ADM, they check the markers they check the markers of those Echinar cells and ADM, and they are able to find out that in the Echinar cells are mostly AMY2 markers are expressed in the Echinar cells and also in the ADM, whereas PRSS1 is expressed mostly in the ADM, and the other three are divided into three different ductals based on their markers, such as this SOX9 is expressed in the three ductals, and KRT19 is expressed in highly expressed in the final ductal, and EPCAM expressed highly in the first three populations or last three populations, ductal 1, 2, 3, all the three populations, and thereafter they check the differentiations using the analysis is shown here, and they can see that Echinar cell seems to be differentiated into the three ductal populations, and through this ADM, or we can say the Echinar cells are finally differentiated into the three different ductal populations, and in between the process this ADM formations is also occur, that it can be seen here, Echinar to ADM, and then it differentiated into different ductal populations, and they check this using the KC and KSC, and they find out that in the case of this KSC, the differentiations seems to be higher for this third ductal cell populations compared to the KC cells. And when they check the using the geo analysis or geo pathway analysis, they are able to see that in the case of ductal 3 populations, they see that upregulation of the inflammatory response and the oxidative phosphorylation response is also upregulated compared to the other cases, compared to the other ductal populations. And when they check the genes that are upregulated, and they can see the genes that related to the EMT pathway, such as twist11, or the differentiation pathway such as KLF, such as this GATA5, KLF5, KLF4 also seems to be upregulated in the ductal 3 populations. And when they check using the GSE analysis that about the oxidative phosphorylation and fatty acid metabolism, they see that the cells that are differentiated from the KSC has more oxidative phosphorylation and fatty acid metabolism compared to the cells that are differentiated from the KC. And once they find out that oxidative phosphorylation is high, and the result of this oxidative phosphorylation is that more oxygen consumptions by the mitochondria occurs. So they check the oxygen consumptions by the mitochondria using this OCR, oxygen consumption rate, and they can see that in the case of this cells that are derived from these KSC tumors has more oxygen consumptions compared to the cells that are derived from KC. These things are quantified in the right hand side of the panel L, where we can see that when the KSC derived tumor cells are used, they see that high OCR consumptions compared to the case where KC derived tumor cells are being used for the OCR. So this further indicates that it's the SETD2 mutations that somehow cause this mitochondrial OXPHOS activations in the pancreatic tumor cells, and these things they have observed also for the other solid tumors, and those results are presented in the main supplemental figures. You can refer to those supplemental figures. So if you need to understand more about this experiments or bioinformatic analysis in detail, just refer to the reference that has been provided in the description. And that's all for this video. Thank you for your time.